Okay, well, let's get started. So today the goal is that we're going to try to go through this homework. And so there's sort of two steps to this. The first step is that I'm going to ask you guys to break up into small groups of 3 to 4. I don't feel like I need to necessarily assign those, but I'm going to ask you to sort of break up into those. And within each of the groups, I want you to nominate a speaker person. And know that if you're a speaker person today, then each of the next 2 or 3 weeks, next 2 or 3 homework things, you cannot be the speaker person. So we're going to try to rotate this around the room. But you'll be in charge of speaking for your group. So what I'd like you to do first is work as groups and just work through each of the different questions, check, basically checking each other's answers and see if you have, if you can come to some consensus on each of the different questions. And then what we'll do is we'll go around the room and after we go through that session, and we'll talk about them as a class. And the person that's the speaker might be tapped to sort of present an answer for a particular question. And then we'll just have a discussion based on that. Okay? So that's the intent for today, and I think it probably will take most of today. So initially, my thought is we'll start with like 15 minutes. We'll see how much time you need. But then basically just work through together as groups, try to get through each question. And then again, we're going to be talking about them as a class. Any questions on that? Okay. Well, with that, go ahead and move around, break into groups. Again, 3 or 4. If it gets much bigger than 3 or 4, it's starting to be too big. Turn off the lights. Okay, good. Discussion. So we're going to try to do that. The first one doesn't work as well for that, but I think we'll just sort of try to put up answers. So we'll start with number 1. Does anybody want to try to do like the first half of it? Just sort of, I'll call out questions and we'll just sort of shout out answers. I don't think I'm going to type in here because it'll get all this stuff. Anybody feel comfortable to go at the first one? Or the first half of this one? Thank you so much. So we'll just start with number 1. So what are these things?
Membrane-bound proteins on a lipid raft, and the bubbly things underneath of them are the hydrophilic heads of the phospholipid bilayer.
Yeah. So did you watch the narrated version?
Yeah, after I watched the not narrated version and struggled.
Yeah, sure. No, that's fine. Yeah, so these are membrane-bound proteins. These could be receptors. I think in this case they're supposed to be cell adhesion proteins of some kind or something. But that's the idea, is these are membrane-bound proteins. This is a lipid raft, which is a term you don't actually have to know for the class. And then these are all phospholipid head groups. And I love the animation because it shows them kind of bubbling around. But you really get the feeling that this is a nice fluid membrane looking at the video. What about... I promise we'll do more of a discussion on those. Number 3, what do you think that is?
That's part of the cytoskeleton. The video said spectrin tetramer.
Yeah, what do you think that is?
But it's there to form the cytoskeleton and provide stability for the cell.
So we have 3 kinds of fibers, right, that we talked about in class. We had microtubules, neurofilaments, which are also intermediate filaments, and microfilaments, which would be things like actin. Any of those fit the bill? Yeah, probably it's some kind of an intermediate filament. Technically, this animation is about a white blood cell, so it wouldn't be a neurofilament, but they're intermediate filaments. So spectrin tetramers are one variety of these intermediate filaments. So they're definitely providing structure to the cell. All right.
That is actin, and it changes the shape of the cell when signaled to do so. It helps to form the microfilament.
Yeah, number 5.
Microtubule, it's the highway of the cell, so motor proteins can move things throughout the cell.
Yeah, I love this video because it's showing the sort of spontaneous way in which these things are formed. And the idea is there's not like some ghostly, well, I don't know, there is kind of a ghostly force. This is driven by, the interactions between these different subunits, and some of them are going to be charged or polar and some are not. And those properties allow them to basically be pushed around quite a bit, and they almost look sort of intentional in terms of construction. This is probably not that far from the truth of how things are actually happening right now within your own cells when we're making actin or making these microtubule filaments. Oops, two more. What's the walking thing?
That is a motor protein, and it's pulling a membrane-bound vesicle walking on a microtubule.
And anybody have any ideas which kind of motor protein it might be? I mean, you would tell based on which way it's going. So that would be the key thing. And like in the next picture, I'm not going to spill the beans on the answer to this one, but the next picture shows these centrioles. So here's the nucleus. These are the centrioles. That's really where all these microtubules are originating. And this vesicle, it's kind of hard to tell, but this vesicle will be walking away from the centrioles, so away from the nucleus. So what do you think? Idea? Yeah, it should be kinase. Whereas dynein would be going the other way. Dynein would be heading toward the centrioles. That's the other kind of protein. Cool. What do you think the organelle is?
Mitochondria.
Yeah, I like them. They look like little, kind of look like a calamari cruiser if you're into your Star Wars. At least to me they do. It's cool to be into Star Wars again, right? So I can come out of the closet on that. I feel like that's totally legit now. All right, let's see. Maybe we'll do one more and then I'll switch to somebody else. So what do you think is coming out of this hole, and what are the holes and all that?
mRNA, and the holes are the pores in the nuclear envelope, which is the membrane.
Yeah. Yeah, so you got little messenger RNA molecules that are popping out. They've already been spliced at this point, so they already would have gone through that process where you cleave out the introns, leave the exons, splice them all together to form this nice messenger RNA molecule that we'll use to do the next thing. So maybe we can, how do groups work here? So you're a 1.
Yeah.
Can we go here? I'll just start rotating around. So who's spokesperson here? Okay, so what did you get for 13? What's the structure?
That was a ribosome.
Okay, so that's a ribosome. And the strand?
You said mRNA?
Yeah, that's the same thing as the prior picture. But here it kind of loops up, and they do this. They kind of loop up in this circle, and then the ribosomes come in, and you'll note there's two parts of the ribosome. There's a large subunit, a small unit, small subunit, and they dock on there. And the actual protein synthesis stuff that happens, oh, I spilled the beans on that, is actually happening mostly within the large subunit. So here you've got the small subunit, large subunit. So let's see. So that's messenger RNA. That's the ribosome still. So what's this thing? Yeah, that's a protein that's being manufactured. So in this case, they're showing it being manufactured, and then it's released in the video. That protein gets released and bound to this little, basically carrier molecule, and it's traveling to this thing, which we already identified as a mitochondrion. I'm gonna let you answer, I promise. But then here's another variant on that theme. So here again, we've got similar structure, ribosome, messenger RNA. What's this membrane?
Endoplasmic reticulum.
So what's happening in this image then?
Protein is being transferred to the endoplasmic.
Yeah, so this here you've got a ribosome that's directly injecting its protein into the endoplasmic reticulum. So this would be rough endoplasmic reticulum, and it's receiving a protein that I think, if I'm trying to remember, I've only watched this video about 1,000 times, but I think in this video it actually shows the protein kind of getting left sort of embedded within the membrane. And if this is going to be a membrane-bound receptor at some point, where the receptor is actually part of the membrane, the receptor would be facing inside to the endoplasmic reticulum. And eventually it would get packaged into the blebs. And then what are those blebs in 21?
Vesicles.
Yeah, those are vesicles, which again could contain receptors facing inward. It could also contain things that are not bound to membranes, and those would be basically just floating inside that little small vesicle. What's that thing?
The Golgi apparatus.
That's cool. So you got the Golgi apparatus, you got all these vesicles coming into the Golgi apparatus, which I just spilled those beans, but we already kind of identified the blebs are the same as these sacs. So they're coming in, they're carrying Will they have vesicles in there? Where are they coming from?
Endoplasmic reticulum.
Yeah, so they just came out of the endoplasmic reticulum. They're carrying protein products that have been manufactured. It's hard to see it, but they're actually being conducted along microtubules, being pulled by those little motor proteins, and they're arriving at the Golgi apparatus where they're going to be sorted and routed, and folding will be completed. What's happening in this picture?
Protein is being transported from the Golgi apparatus to the plasma membrane.
Yeah,
in a
so
vesicle.
there's the plasma membrane up there. You can see those spectrin tetramer intermediate filaments that are up there being held onto by an actin molecule, but here's a microtubule routing them up there. And then finally you get to the surface, and what's happening here?
Proteins are being secreted from the cell.
So here you got protein release, and then also like a screen grab later, which I didn't get, you see the membrane-bound proteins that were inside that vesicle. Once the membrane fuses, they then are now they're being displayed outside the cell. And these things can function as receptors or cell adhesion proteins or whatever it is they are. Any questions on any of those things? I love that video. If you did that in Bio 111, if you did the same worksheet at some point, it's possible because I gave all this stuff to other people. But I just think it's such a good way to walk through that process of gene expression and then protein synthesis and how the different parts of the cell are involved in that. I think it's a just beautifully done thing. I remain convinced that there's at least a 50-50 shot that if that would have existed back in my day when I was an undergrad, those many years ago, I probably would have been a cell biologist because I feel like it would just grab me. But didn't happen. I went behavior instead, and I'm very happy for it. But I really like that video. Okay, so number 2. Explain the figure shown in box 2.2, page 33, which tries to explain how one can use microarrays to look at differences in gene expression. So we've done that. Is this a group there? E3. So maybe you guys could sort of walk us through this one. So what's happening and what's in these 2 vials that are, you can't see it in the monitor, but that one's red, that one's green.
So for A, said that on both the vials you define mRNA. And as it relates to gene expression, that the mRNA will undergo the translation, which creates proteins. And it's these proteins that carry out the specific functions and dictate what cells can and cannot do, which results in gene expression.
Okay.
For part B, we just said that in the plate, small spots of synthetic DNA are on the plate before the mixture is applied. The small spots of synthetic DNA contain a specific DNA sequence that will recognize a specific mRNA sequence. And then part C, so once the vials are applied and mixed in the plate, different colors appear. The different colors appear because the DNA will bind together with its complementary base pairs of mRNA. And then the colors appear just basically what the picture says.
Right. I guess it does kind of just say it, doesn't it? So what does red mean then? Before I can find it.
Gene with reduced expression.
Yeah, in brain 2 it says. So if it's let me just step back. So before I even step back, because I'm going to just start talking. So any differences in the first 2 or other things people would say? I think that's pretty much dead on. So you've got these plates, and each one is manufactured such that it has a specific sequence of DNA in it. It would be many strands of DNA, but they're all the same sequence. So this one, for example, could be ACTGGC, and then they're probably longer than this most of the time. This one might be TGCCGA, so that I'm giving you 2 codons per DNA sequence. And basically what you're looking to find is if RNA in one or both of the brains binds to that DNA. So if you had an RNA strand from the red brain that ran U, A, G, C, C, G, yeah, that RNA strand would then interact with your cDNA. And the key thing is these messenger RNA strands, these have been labeled with some kind of a red dye. So anytime you get brain number 1's messenger RNA binding to the DNA, the little well will start to appear red because it's got red labeled messenger RNA in it that's sticking to the DNA that's stuck to the plate. Whereas the green brain, if there's no green messenger RNA that also adheres to that well, then the green will just sort of wash out after you apply it. It won't stick, and therefore the well will appear red and not some other color. And then in contrast, if you happen to have messenger RNA that was labeled green that matched up to a different cell, so here it would be, no, not T, U, C, G, G, C, A, no, U. Not that I don't know this, it's just that it's in front of people. In that case, the thing might appear green. So anything where the well appears red, that means that the figure says that it has reduced expression in brain 2, but it also could mean that it has increased expression in brain 1, which is what was labeled red in the vial, right? So the red vial up top shows from brain 1 labeled red. So anything that's red, that means you're making it in brain number 1. Whereas anything that's labeled green, you're making that in brain number 2, but not in brain number 1. At least not at the same rate. Does that at least make sense? What happens if it's yellow? Like, what does the yellow mean? Yeah? Yeah, that's what it's supposed to be. So if you got, if you had other strands, I'm not going to write them out, but if you had another strand, I'll do it. So if you also have DNA strands in there that are, they're the same DNA strands, but if there's the same messenger RNA strand that's from the red brain that's also being produced, it could also bind GCU. And in that case, you'd have both red dye and you'd have green dye in the same well. And under that scenario, basically that means that there's equal amounts of red dye and green dye, so it would appear not red nor green, it would appear a different color. And they're claiming it would appear yellow here. Does that make sense? Yeah?
Is it common to use the same well plate for 2 different samples?
Yeah, so this is one of the real powerful things of this, is that by applying 2 different samples to the same well plate, you can get an idea of how gene expression differs between these 2 brains. Because now we know, because we bought the plate and the manufacturer told us what cDNA, what complementary DNA is found in each of the different wells. And so looking through here, we can say, hey, this thing has a lot of, brain 1 is expressing more than brain 2 on that particular sequence. And brain 2 is expressing more than brain 1 on that particular sequence, the green ones. And so by basically comparing those 2, we all of a sudden have an immediate indication of what differences in sequence are present between those 2 brains. So that's one of the real powerful things about this technique. And you could be screening, these wells might have 100, these plates might have 100 different wells on them. And so you're able to screen for effectively like 100 different genes and see how those genes might vary in expression between your 2 samples. It's kind of a neat technique. So you can sort of imagine, like, what if one of these brains came from an autistic person and one of them came from a non-autistic person? We're trying to figure out what's different in gene expression between brains of autistic people versus not. This is going to allow us some very initial screening. It's not going to be conclusive about, like, the fact that that's red doesn't mean that that's the gene for autism. But it does indicate that there's a difference there that we could then investigate and see its impact by doing further studies. Yeah?
So what would happen theoretically if there's a certain gene that turns a different color, but that gene can also indicate another type, not just like, I don't know if that's making sense. Like a certain gene can be a gene for 2 different types. How do you differentiate between which one?
Yeah, I mean, all this will tell you is for the particular complementary DNA sequence that is present in that particular well, which brain is expressing more of it. So if I'm understanding your question, if the thing that differs between the genes is not sort of contained in that part of the sequence that's in the well, then it won't appear in the assay. Not sure if that's quite what you're saying, but so like if the gene is found, if the gene's 100 nucleotides long, and we're only assaying 20 of them in the well, then if the difference between the two brains is not contained within that sequence, it won't affect, it won't show up in this assay. I think hopefully that's helping. It's a neat approach. It's something that allows us to do sort of a whole-scale, quick comparison of gene expression, in a very short period of time. Anytime we do this, we'd want to do further studies afterwards. But this might allow you to go from not being, having 100 genes you're potentially interested in, down to maybe having 10 genes that you're really interested in, or 20 genes. And so therefore, then you can pursue other techniques to really hone in on those ones that are lighting up as potentially being different. That's the idea. Let's finish saying this. The thing that I remember, I've been asking this question or some variant on this question either as part of lecture or as part of homework for years. And I think it was last year someone pointed out that red and green don't make yellow usually. and this goes back several editions of this book too. So but I'm convinced that really the green should probably be yellow and the yellow should be green in the figure, and it would make more sense, kind of, because it's dye. It's just dye. I don't know. But the point is the same, is that you're basically tracking and you're looking for, same color versus some different color when you're comparing across these things. The other thing that it sort of indicates, I think, is that if you want to go into this work, you probably need to not be red-green colorblind. So my undergraduate molecular biology instructor was red-green colorblind, and he always told about trying to do these assays, and he's just looking at the thing, he's like, I don't see any differences at all. All right, number 3. Alzheimer's disease. So, let's see, we went there. Can I go over here? Is there a group sort of right in this area? Who's this? Is that you?
That was us.
You were there, right. Well, we'll go. That's great. Okay, so we'll do the next group. So let's start with tau protein. What's normal function of tau protein?
It stabilizes the microtubules and makes sure that they stay parallel and straight.
Okay. And what's good for B? What happens during Alzheimer's disease?
The tau separates from the microtubules, causing them to fall apart, and then the strands that fall apart combine and form tangles that destroy the cell.
So what did you say about specific function of cytoskeleton?
Cell won't work if the cytoskeleton is destroyed.
Is there anything different or have additions to that they wanted to make? I think it's pretty much spot on. So tau's this interesting stuff in that picture, and it's hard to see up here, but you've got it on your screens and you saw it in the video. I mean, it basically is functioning kind of like tape. And so the tau protein, as I sort of imagine it, holds all these little dimers that form the structural, the key structure of the microtubule is formed by these little tubular dimers. But the tau basically holds them all together. And so what happens during Alzheimer's disease, apparently somehow related to the fact that you're also accumulating this other protein called beta amyloid. And the connection is not, I have not seen really clear connections, but there does seem to be a connection between the two. As beta amyloid accumulates, that causes tau, it kind of ran off the bottom of the page, but it causes tau proteins to become hyperphosphorylated. So on your thing, hopefully the image still shows the hyperphosphorylated tau, where just you start to have little phosphate groups attached to the tau protein. That causes them to lose their affinity for the microtubule tubulin dimers, and at that point, they no longer can hold the microtubule together, and the thing just blows apart. And the tau then instead forms these little tangles that are found within neurons. I think it's a little up in the air whether the fact that there's these there are these tau protein tangles present within the neurons, if that is actually destroying the cell, or if it's more just like an indication that the cell is being destroyed. Because it's not entirely clear to me that those tau protein tangles actually damage the cell per se. But the only time you ever have those tangles is when your microtubules have fallen apart. So that's the bad thing that's happened. And now, and so as we talked about, if you don't have microtubules, you no longer have any kind of a good, efficient mechanism to move materials from one side of a neuron to the other. So here's my little drawing of a neuron. That's how I tend to draw neurons. And here's a little motor protein that's supposed to walk all the way down that microtubule down to the axon terminal. And if you lose your microtubules, it can't do that anymore. And so as a result, you have axons dying within those neurons.
Did you say they become phosphorylated? That's why they...
Yeah, they become hyperphosphorylated. And there's other cool things about it. Like, apparently, if you have hyperphosphorylated tau present at all, that hyperphosphorylated tau itself can induce other tau to become hyperphosphorylated. So in that way, it kind of functions, well, it functions in like a little positive feedback loop where you have an initial little bit that happens, and that causes more to happen, which causes more to happen, which causes more. And the other thing about that is that it, in that way, also tau, hyperphosphorylated tau anyway, is functioning as a prion. It's kind of like an infectious thing, because once you have hyperphosphorylated tau present, it can catalyze the formation of more of it. Similarly, the beta amyloid has been identified as a prion. So there's a way in which Alzheimer's disease may be basically a function of these two prions that are causing the molecular level changes that are happening within the cell, the beta amyloid prion and the tau prion. There's a good review article I have. I'll try to post it. It's called, the title is A Tale of Two Prions, and it goes in and discusses these two, the function of these two proteins in causing Alzheimer's. That's close to what I want to say. Questions on Alzheimer's disease? It's a good topic. All right. I don't know if we have time for this or not. Yeah, let's try. So number 4. Is there a group in that back corner? Okay. So what did you put for number 4?
This is the article suggests that Addison motors are used for the anterograde travel to the plasma membrane and going into the rest period for the return portion of the trip. So the Kinesin 1 motors are used for multiple voyages to the plasma membrane, suggesting that they aren't removed after a single journey, but ultimately out during the retrograde portion.
Right.
They're used again on later anterograde travels.
Yeah, so here's the, I mean, this is the gist, I think. It's not that they kind of run all the way to the end and then they go down there and they just get destroyed. That was one hypothesis, and that doesn't seem to be the case. Instead, what seems to happen is they go part of the way down, and then at some point, they seem to just kind of break off and from their vesicle, from the microtubule. They leave the vesicle behind, and then another one will swoop in and take it another segment of the way down. And then they'll kind of trade off and have another one and another one. And each time, the kinesin then just kind of diffuses. I mean, it would diffuse in either direction, but on a net basis, it would be diffusing back up the axon. And, so this one that broke off here might come back and then grab onto another vesicle. So they kind of seem to do a loop, and they take turns, almost like kind of a bucket brigade. They kind of travel a ways down, let go, break off, and then another one will take over and take that vesicle on. And then they will pick up again and carry it all the way down. this stemmed from a student question that happened in class. They're like, well, what happens to kinesin when they hit the end? I'm like, I don't know. So I looked it up. And there was this paper that had been published like 6 months earlier at the time. So I think it's kind of a cool thing. That seems to be what happens. They just keep reusing these same proteins over and over again. Okay.